Bonjour à tous. Merci beaucoup pour votre présence pour cette deuxième conférence midi de l'année du CRI-UGM. Ça fait plaisir de pouvoir voir qu'il y ait du monde du connecté et qu'il soit toujours là, fidèle au poste. Merci beaucoup. Bon appétit pour ceux qui sont sur la pause du lunch. On en profite, c'est l'avantage du télétravail. Quelques informations pour vous. Les règles par rapport au chat sont indiquées, par rapport aux caméras et par rapport au micro sont indiquées dans le clavardage. De manière générale, on vous demandera d'avoir vos micros coupés, sauf si vous posez des questions. Et comme les bandes passantes s'améliorent avec Zoom et que les enregistrements de la caméra sont faits, on vous propose, pour ceux qui le veulent, d'avoir leur caméra d'activer si vous le voulez, ce qui rend la présentation plus vraie et plus user-friendly, plus sympa. Et donc ça, c'est la première chose, sachant que si jamais vous avez un problème de connexion, la présentation sera en ligne derrière. Mais normalement, on n'aura pas de souci par rapport à ça. On veut garder l'aspect vraie vie et l'aspect interaction au maximum. Je m'excuse, je vous présente mon chat également. Donc voilà, c'est le télétravail. On va y aller, c'est parti. Aujourd'hui, on a le plaisir d'accueillir Dr Sylvia Santosa de l'Université Concordia. Dr Santosa a obtenu son doctorat en sciences de la nutrition à l'université McGill, puis a effectué ses recherches postdoctorales à la clinique Mayo aux États-Unis. Elle est aujourd'hui professeure associée au département de santé, de kinésiologie et de physiologie appliquée de l'université Concordia. Sylvia est également chercheuse au centre Perform. À l'hôpital Sacré-Cœur, elle est aussi chercheuse associée au CRI-UGM. Dr Santosa est également titulaire d'une chaire de recherche du Canada de niveau 2 en nutrition clinique. En tant que titulaire d'une chaire de recherche du Canada en nutrition clinique, Sylvia mène des recherches de pointe pour identifier les effets à long terme de l'obésité, de la cellule à l'organisme tout entier. Ses projets actuels sont les suivants. Oups, j'ai un problème d'écran. Voilà, excusez-moi, désolé pour ça. Ses projets actuels sont les suivants. Examiner les différences régionales dans le tissu adipeux des patients ayant subi une chirurgie bariatrique. Investiguer les différences entre l'obésité précoce et l'obésité tardive. Déterminer les besoins en protéines des patients ayant subi une chirurgie bariatrique et l'utilisation de techniques d'imagerie pour examiner l'utilisation régionale des graisses et l'oxydation des substrats. Comme vous avez pu le voir dans le résumé, la présentation va être en anglais. And Sylvia, we're going to switch in English for you, and I will let you keep going with your presentation and with the title that will be better said with you than by me, with my accent. So have fun with your presentation, and thank you again for accepting to present your work with us. Thank you a lot.
Thank you very much, Ben. I am just going to share my screen and start my presentation. I just have to change these little boxes here. Oh, there we go. Okay. Okay, so thank you all for joining me today. I'm going to be talking to you about subcutaneous adipose tissue, especially in the femoral area, which is generally an unappreciated depot, but can be a key player in metabolic disease risk. Oh, wait, let me get my pen out, my little laser pointer. Can you see it? Okay, so there are various depots in the body, and my research program aims to examine the factors that affect these various depots, and then to delineate what the effects are of these depots on disease risk. Though there are a number of fat depots in our body, what I would like to talk about or focus on today is really the visceral fat depot, as well as the upper body subcutaneous fat depot and the lower body subcutaneous fat depot. I'm going to show you that not only is the visceral depot different than the subcutaneous depots, which we already know and is already well demonstrated, but I also want to demonstrate how upper and lower body fat are significantly different. In doing so, I will also show you the importance of lower body fat, an underappreciated depot in the development of obesity-related comorbidities. So we know that not all obesity is equal. There's a range of body shapes, and at either end of the spectrum, we have those that tend to be apple-shaped who preferentially store fat in their upper body, and those who tend to be pear-shaped that preferentially store fat in their lower body. Now, a while back, I did a talk for CRI-UGM, and I spoke about how the comorbidities of aging, such as cancers, cardiovascular disease, gastrointestinal disorders, type 2 diabetes, etc., actually mirror those of obesity. And since then, we've actually published an article that looks at how, or more closely examines how obesity mirrors aging. What I wanted to talk to you about today is that also it is well established that those who tend to be apple-shaped and store fat in their upper bodies are at actually greater risk of these obesity-associated comorbidities. In fact, as you can see from this figure, we have mortality hazards ratio on the y-axis and BMI on the x-axis. And what you can see is that regardless of BMI category, there's a greater risk of mortality in those in the highest waist circumference category compared to the lowest waist circumference category. And if you look a little closer, you can actually see that those within the normal range of BMIs have a greater risk of mortality than those who are classified as obese or in the obese BMIs and have a lower waist circumference category. In fact, those in the highest waist circumference category who are lean actually have a similar risk to those who are in the highest waist circumference category who are obese. Because of this, many studies like this, a lot of research has been conducted to examine the pathogenic effects of upper body fat. This is a figure from one of the landmark studies, this one being from the European Prospective Investigation into Cancer and Nutrition. This figure shows that in addition to BMI, waist circumference and waist to hip ratio is associated with the relative risks of death in both males and females. Notice here that a lower waist to hip ratio appears to be related to a lower relative risk of death in comparison to the BMI and waist circumference curves where you have a little bit more of a J. Thus, though BMI and waist circumference and waist to hip ratio are all equally associated with the relative risk of death, waist to hip ratio may be more sensitive to predicting risk as the relationship is less J-shaped. Furthermore, in this smaller study, it shows that waist to hip ratio is the strongest independent predictor of cardiovascular disease death compared to BMI and waist circumference. So in these graphs, all-cause mortality ratios are on the y-axis, and on the x-axis, we have the hip circumference, the top graph being in men, the bottom graph being in women. Here, I would like you to focus on the solid black line, which is the adjusted line for smoking habits and waist circumference. And as you can see from these solid black lines, opposite to waist circumference, hip circumference is actually negatively associated with the rate ratio of all-cause mortality in both men and in women. This begs the question as to whether it is actually greater upper body fat or less lower body fat that is actually driving the risk of mortality. Now, as many of you know, most of the studies that compare abdominal subcutaneous and visceral fat show that visceral fat is particularly pathogenic, with greater fat cell size, immune cell infiltration, and secretion of several inflammatory cytokines. This is further illustrated here, where you can see that as obesity develops, differences in pre-adipocyte cell characteristics cause subcutaneous adipose tissue to expand more by hyperplasia. So you get greater proliferation and then differentiation here. And compared to the omental or visceral fat depot, where you don't have the same ability to proliferate, and then you ended up with fewer but larger fat cells. Now, it's significant when the fat tissue expands by hyperplasia versus hypertrophy, because larger adipocytes are thought to be more pro-inflammatory as they are more prone to adipocyte death and the subsequent recruitment of immune cells, especially macrophages. It's also interesting to note that femoral subcutaneous adipose tissue actually has a greater ability to replicate and undergo adipogenesis and become more numerous in studies where overfeeding occurs. So this is all well and good, except that at any given weight, subcutaneous adipose tissue makes up approximately 80% of fat tissue in the body. So in comparison, visceral adipose tissue makes up 10% of fat in lean men and 5% of fat in lean women. So you actually consider the relative quantities of visceral to subcutaneous fat, though per gram visceral fat is more pathogenic. The relative amounts of subcutaneous fat in our body makes it an arguably important player in obesity-related pathogenesis. For example, there are more overall immune cells in visceral adipose tissue, as you can see here on the left, compared to subcutaneous, or upper body, I should say, subcutaneous adipose tissue. And proportionally more inflammatory versus anti-inflammatory cells in visceral compared to upper body subcutaneous adipose tissue. So here you can see the anti-inflammatory cells are in blue and the pro-inflammatory cells are in red. However, something to consider is that at any given weight, there is likely to be 8 to 16 times more subcutaneous fat than visceral fat. Subcutaneous fat, especially in the lower body, is thought to be a safer place to store lipids. Our view is actually that femoral and abdominal adipose tissue expands as I should say, femoral and abdominal subcutaneous adipose tissue expands. These depots become dysfunctional. So they end up with some impaired characteristics that have not yet really been identified very well. And as a result of this dysfunction, the subcutaneous adipose tissue directly contributes to metabolic disease risk and also indirectly contributes to metabolic disease risk by increasing visceral adipose tissue, which then contributes to an increase in metabolic disease risk. So there's much evidence out there in support of this hypothesis. Here is a before picture on the left. Okay, and then on the right is an after picture taken 6 months after liposuction. So this is liposuction of the abdominal subcutaneous fat. And as you can see, 6 months after liposuction of abdominal subcutaneous fat tissue, there was a compensatory increase in visceral fat. On average, visceral fat in this group increased approximately 10%. So this increase in visceral fat supports the idea that when fat cannot be stored in the subcutaneous adipose tissue depot, it accumulates as visceral fat. And not all subcutaneous fat is actually the same. This figure illustrates fat metabolism in grams per day for a normal weight male and female. Here, I'd like you to focus on the red arrows that indicate the amount of fatty acids that are being contributed by each depot to the circulating free fatty acids. Now, as you can see, the red arrows differ significantly between depots in men and women. So the upper arrow indicates the contribution of upper body subcutaneous adipose tissue. And as you can see, upper body subcutaneous adipose tissue actually contributes the majority of over 60% of circulating or systemic fatty acids. So in women, it contributes 125 grams per day, an estimated in lean women. And in men, it's estimated that there's a contribution of 138 grams per day from upper body subcutaneous fat. In contrast, visceral adipose tissue contributes 17 grams per day and 27 grams per day to circulating fatty acid concentrations. Also, if you look at lower body depots, the contribution in lean women averages about 43 grams per day, and in lean men, it's about 28 grams per day. This is equivalent to approximately 15 to 20% of circulating free fatty acid release. Now, what I want you to take away here is that abdominal and femoral subcutaneous fat are different. And that free fatty acid release from the subcutaneous bed actually accounts for over 75% of systemic free fatty acids and is likely a major contributor to lipotoxicity as adipose tissue expands in obesity. The abdominal fat is actually the largest contributor to circulating free fatty acids. Femoral subcutaneous adipose tissue is an underappreciated depot that likely plays an important role in the pathology of obesity. In this double-blind PET study by Virtanen and colleagues, participants were randomized to receive metformin or rosiglitazone treatment for 26 weeks. And here I'd like you to focus on the rosiglitazone treatment, which is in this last light gray bar. Now, what's interesting in this study is that with the decreases in visceral fatty fat mass, so you have a decrease in visceral fat mass after that 26 weeks with rosiglitazone treatment, there was an increase in glucose uptake, okay? And then, but not, it was, the increase in glucose uptake was in the per kilogram depot of adipose tissue, but not in the whole fat depot. But if you look to the right, there didn't need to actually be changes in femoral subcutaneous fat mass in order for there to be huge increases or relatively larger increases in glucose uptake in the per depot and the whole body adipose tissue. Moreover, so this is an adipose tissue, but femoral adipose tissue is actually highly correlated with whole body glucose disposal rates as well. There's another study here by a group in Quebec that I wanted to show you, which supports the importance of the role of femoral subcutaneous adipose tissue in obesity-related comorbidities. These investigations looked at how changes in body composition corresponded to the resolution of hypertension and obstructive sleep apnea. So in this study, there were 62 bariatric surgery patients that were included. And to measure body composition, they examined body composition via bioelectrical impedance analysis and computed tomography at baseline and at 12 months post-surgically. Here, they looked at the individuals who had unresolved hypertension and the changes in body composition. So as you can see, in those who had unresolved hypertension after 12 months, there was actually a lower reduction in visceral adipose tissue, but there was also a lower reduction in the mid-thigh subcutaneous adipose tissue. Also, in those who had unresolved obstructive sleep apnea, there was a lower reduction in mid-thigh subcutaneous adipose tissue only. So this study actually supports the notion that changes in femoral subcutaneous fat drives the resolution of comorbidities after bariatric surgery. There are actually very few studies that compare the characteristics of femoral subcutaneous adipose tissue to other tissues in other depots to see how femoral fat uniquely contributes to obesity-related comorbidities. We used a flow cytometry technique that we recently published to examine the differences in immune cells in abdominal and femoral subcutaneous adipose tissue. For those of you who haven't done flow cytometry in adipose tissue, or even worked with adipose tissue, it is not an easy substance to work with because adipose tissue is oily and the oil residues just stick on everything and it's really hard to get rid of. And most of the solutions that we use are aqueous solutions, and of course, adipose tissue is hydrophobic, and so we have to find ways around that. So here on the left, I'm showing you what we could have hoped for before we perfected this flow cytometry technique. And what you can see, this is 1 gram of adipose tissue, which was considered to be the minimum. So those who did animal studies with flow cytometry, what they would do is they'd have to pool the adipose tissue from various different mice in order to get that 1 gram, because you wouldn't get 1 gram of adipose tissue from just one mouse. So as you can see from this image, it's pretty messy. There's a lot of autofluorescence. There are not clear populations. Now, this is colored with the actual computer program, but if you picture all of this in red, you can see that there's not really very clear cell populations. All right. And then on the right, this is our improved technique, and we can consistently get these clear cell populations with pretty much no autofluorescence. So as you can see here, there's a very clear population up here, and there's also a macrophage population, and this is our lymphocyte population down here. So we can get these clear populations with approximately 500 milligrams of tissue or less. It took us a long time to perfect this technique, and using this technique, we are able to better understand the immune cell profile in regional adipose tissue depots and what affects them. This is important because adipose tissue immune cells are important contributors to inflammation in obesity. Now, our paper that is currently in press in obesity has found preliminary differences in immune cells between abdominal and femoral subcutaneous adipose tissue. As in the previous diagram, the anti-inflammatory cells here are marked in blue, and the pro-inflammatory cells are marked in red. In females, we found that there was a greater proportion of pro-inflammatory T cells in femoral than abdominal subcutaneous adipose tissue, and that the proportion of M1-like macrophages were actually greater in abdominal than femoral subcutaneous adipose tissue. We have also looked at markers of DNA damage and cellular senescence, which typically occurs in response to DNA damage, and is a state in which a precursor cell ceases to proliferate and differentiate. So basically a cell cycle arrest. Once a cell enters a senescent state, it is irreversible. And in the senescent state, the cell will adopt a pro-inflammatory phenotype known as a senescence-associated secretory phenotype. The number of senescent cells increases with both aging and with obesity. This is our unpublished data, and our unpublished data show that at the whole adipose tissue level, there is a greater cellular senescence in femoral compared to abdominal subcutaneous adipose tissue of individuals with obesity, and these are in individuals that are otherwise healthy. So the only thing that they have is obesity, and they're non-smokers. Okay, so you can see that there's a greater amount of beta-galactosidase positive cells, which indicate cellular senescence. So this indicates that cellular senescence may be a source of inflammation in this depot. Interestingly, in pre-adipocytes from femoral subcutaneous fat, P21, which is a transient marker of cellular senescence, is lower than in pre-adipocytes from abdominal subcutaneous fat. P21 expression usually goes down after the establishment of growth arrest, so this is in line with our beta-galactosidase staining. Acetylcoa is a central metabolite that has increasingly been implicated in blood glucose control and type 2 diabetes, and is actually generally underappreciated for its central role in metabolism. In those with diabetes, there appears to be a downregulation of genes associated with acetylcoa network in adipocytes, implying metabolic dysregulation. Acetylcoa is generated from the catabolism of macronutrients in the mitochondria. So you could, as you know, you could generate acetyl-CoA from glycolysis of carbohydrates, from beta oxidation of fats, as well as from the breakdown of conversion of proteins, or amino acids, I mean. In the fed state, acetyl-CoA condenses with oxaloacetate to form citrate. The citrate can then be exported from the mitochondria. So the citrate can either continue to undergo the Krebs cycle and then be converted to other metabolites in the Krebs cycle, or it can be exported like it is in the fed state from the mitochondria to the cytosol. In the cytosol, this enzyme called ACLY, also known as the ATP citrate lyase, can then turn citrate back into acetyl-CoA, and this acetyl-CoA can then undergo de novo lipogenesis and acetylation reactions. In mice, low ACLY or ACLY deficiency has been shown to result in insulin resistance. Recently, we published a paper in Endocrinology that examines the markers of mitochondrial function and acetyl-CoA metabolism in females with childhood onset obesity compared to adult onset obesity. Though the differences we found in subcutaneous adipose tissue between childhood onset and adult onset obesity are outside the scope of this talk, I would like to show you some of the data here, as these data also show differences in abdominal versus femoral subcutaneous fat. Here you can focus on comparing the white bars to the red bars. So the white bars being abdominal subcutaneous adipose tissue, and the red bars being femoral subcutaneous adipose tissue. And in this figure, we're looking at acetyl-CoA concentrations or levels. And this acetyl-CoA level is representative of cytosolic acetyl-CoA. So as you can see in this figure, the cytosolic acetyl-CoA in femoral subcutaneous adipose tissue is significantly lower than abdominal subcutaneous adipose tissue. Now, comparing the red and white again, you can see that the lower cytosolic acetyl-CoA levels in femoral SAT over here in the red correspond with is actually lower than the abdominal ACLY levels. And this is in females with obesity. Okay, so this is lower than in the white bars. And our data also show that ACLY mRNA is strongly predictive of levels of cytosolic acetyl-CoA. So these correspond with the increase or the higher acetyl-CoA, I should say, the lower acetyl-CoA levels in femoral subcutaneous adipose tissue from the previous slide. A reminder that lower ACLY has previously been associated with insulin resistance in female mice. A low NAD plus to NADH ratio is also associated with mitochondrial dysfunction. And as you can see here in comparing the white and red bars, the abdominal subcutaneous adipose tissue has a lower ratio of NADH or NAD to NADH compared to the femoral subcutaneous adipose tissue, indicating greater mitochondrial dysfunction in the abdominal subcutaneous adipose tissue depot. Also, there was greater pro-inflammatory IL-6 mRNA expression in the femoral subcutaneous adipose tissue depot of these individuals compared to the abdominal subcutaneous adipose tissue depot. And another small study conducted by Devu et al. Also showed that in overweight females, genes associated with immune cell chemotaxis and inflammatory pathways were particularly upregulated in lower body fat of those who had an upper body fat distribution. So through this talk, I hope that I've shown you that in addition to visceral fat, subcutaneous fat is the largest adipose tissue depot and is a potentially important contributor to metabolic disease risk. Dysregulation of subcutaneous adipose tissue contributes to an increase in visceral adipose tissue and increases metabolic disease risk. And that abdominal and femoral subcutaneous fat characteristics are distinct. Lastly, I hope I've shown you that femoral subcutaneous fat may have a unique and important role to play in mediating metabolic disease risk and is a depot worth further investigation. Thank you very much for your attention. And I would like to give you my acknowledgements or present my acknowledgements. Bjorn Tam and Jessica Murphy were the students that did a lot of the studies that I presented today. Carrie, Delaney, Jessica, and V were instrumental in helping us to perfect our flow cytometry technique.
Thank you so much, Sylvia. Merci beaucoup. Thank you so much for this very interesting talk. Je vous propose pour les questions de poser des questions en français ou en anglais, ne vous gênez pas. On pourra se débrouiller pour traduire. So don't hesitate to ask questions in French and English. We're going to manage to translate if needed. So that's the first thing. Don't hesitate. And the room is open for questions. So don't hesitate to ask your question. Mylène?
Yeah, hi Sylvia, thank you for your talk. It was an amazing and interesting talk, and mostly about the femoral saturated subcutaneous adipose tissue, which is not often studied like you did. But my question is more about epidemiologically or in regard of the population. Do you think if you have a huge amount of femoral subcutaneous adipose tissue during your childhood or your teenager, because genetically you are predisposed to be a peer and not a helper, is it changing something about the metabolism of your adipose tissue compared to somebody who become a peer during the aging process, like a menopause?
Oh man, that's an amazing study that we'll have to do. I don't think we actually know that. We're only at the stage right now as looking at overall like childhood versus adult onset obesity and what the effects are. So these are individuals who've had obesity since childhood or obesity since adulthood who are now adults. So we didn't do any longitudinal sort of thing. It was just like a retrospective thing. And we do see differences that are interesting between the childhood and adult onset obesity. And it seems that those with childhood onset obesity have a greater risk of the various metabolic diseases, including, you know, like they have, for example, a 6-time greater risk of, I think, diabetes, or is it 12 times, something like that. It's much greater. And so it is an interesting population to compare. And we already know that, for example, in childhood onset obesity, there are more numerous adipocytes compared to adult onset obesity, where they have more hypertrophic, larger adipocytes. But as for the actual regions, I don't think anyone's really looked at. So is it, if you're a child and you develop more your adiposity in your lower body compared to your upper body, is there going to be a difference in your disease risk? I mean, that would be a very interesting question. And we do have body composition data in our individuals. So we've done whole body DEXA scans as well as single slice CT scans, so we can delineate the visceral adipose tissue depot as well as the upper and lower body adipose tissue depots. And so maybe what we'll do is we'll take a closer look and see if there's relationships between those who tend to be apple and pear shaped in childhood, who've had childhood versus adult onset obesity. So it's a very good question, very interesting. We'll definitely look towards addressing that.
Thank you.
Hélène?
Yes, hi, a very, very interesting presentation. My question is about, you know, the link between inguinal and abdominal obesity. Does it change in function of the woman's status, for example, breastfeeding, pre and post-menopause?
I'm sorry, did you say visceral, inguinal?
Abdominal
like in mice?
Inguinal, or visceral versus inguinal fat? Or maybe I should say the femoral versus abdominal.
Okay, like your visceral, does it change in breastfeeding?
I mean the relationship in between these two regions in terms of inflammatory profile, does it change in function of the hormonal status of the woman? For example, we know that when a woman is breastfeeding, the visceral fat is not a cardiovascular risk. So probably because there's a, you know, inflammatory profile that is decreased. I don't know. We don't know the mechanism, but I'm interested to know whether you know whether what you observe in obese women, whether this change in function of the hormonal status.
Yes, thank you for your question. That's a very interesting question. And I actually do, I guess the advantage of working from home is I have actually slides that I can show you what we've done previously is look at hormonal status and dietary fat storage. I'm just going to bring that. Let me see if I can bring that up. One second. Hopefully it's in this. Oh, here. Okay, here. So we previously did this study while I was a postdoc, when I was a postdoc at Mayo. And what we did was we looked at chronic estrogen deficiency. So we compared postmenopausal and age-matched premenopausal females, and then also acute estrogen deficiency. We use Lupron to suppress estrogen acutely. And then we had a group where it was a positive control where we added back estrogen. And then in males, we looked at individuals who had chronic testosterone deficiency because they had chemical castration for greater than 6 months, and to age-matched males. And we also looked at acute testosterone deficiency, where we also implemented that Lupron intervention to suppress testosterone levels for an 8-week period. And what we saw was there was actually effects in terms of chronic estrogen deficiency on meal fat oxidation, fat oxidation, and lipoprotein lipase, which is enzyme involved in lipolysis, and then as well as free fatty acid storage and adipogenic protein. So in chronic estrogen deficiency, we saw increases in meal fatty acid storage in total and regional fat, and decreases in fat oxidation, as well as greater LPL in abdominal, the abdominal depot from the fed to the fasted state. And then we also saw that there was greater storage of circulating free fatty acids in the femoral compared to the abdominal depot, which may have been facilitated by various adipogenic proteins. But then you see in the acute estrogen deficiency, there aren't any differences. So you do see differences in some regional effects in chronic estrogen deficiency, but not so much in acute estrogen deficiency. And then in chronic testosterone deficiency, you can see that there are greater meal fatty acid storage in the femoral region and less fat oxidation. But then there's no differences in LPL, and then no differences in systemic free fatty acid storage. And then there's this Acetylcoa synthetase may play a greater role in testosterone, in storage in testosterone sufficient males. And then in acute testosterone deficiency, you can see that there's still that increase. Even when there's an acute testosterone deficiency, you see an increase in femoral fatty acids, meal fatty acid storage, and not in abdominal subcutaneous meal fatty acid storage. So we do see differences with hormonal changes or sex hormone changes, at least in meal fatty acid and fat oxidation, as well as these other outcomes that we looked at. Whereas as for breastfeeding, I'm not very sure in terms of how that might affect things. There's a lot of questions still to remain, like what happens in pregnancy in terms of expansion of adipose tissue as well.
I also wonder, did you try to make a correlation with the morphology, genoid versus android morphology?
Yeah, so in our studies, we haven't done that in terms of the studies that we've published yet, but we will be doing that for some other data that we'll be publishing soon. So certainly something to look at. Thank you for your suggestion.
Okay, thank you very much. Very interesting.
Yeah.
Merci beaucoup pour ces questions vraiment très intéressantes. Éléonore, tu as une question pour nous?
Yes. Thank you, Dr. Santosa. Your presentation was pretty interesting. In fact, during my PhD, we did an exercise training and look at the inflammatory profile of femoral, subcutaneous adipose tissue versus abdominal. And we were surprised to observe no change in abdominal tissue compared to subcutaneous, the femoral one. In fact, in the femoral adipose tissue, we observed an increased mRNA level of interleukin-6 and TNF-alpha. So I always wonder if, you know, because you suggest that femoral adipose tissue should also be considered in the development of chronic disease, and there are also hypotheses on the fact that this tissue can be protective. So
Yes.
we have like two sides, and I wonder if it's a really good idea to favor mobilization of femoral adipose tissue when we don't really know how it will react in terms of inflammation or stuff like that. So I don't know if you have the answer, but you know, it's interesting to see how you look at the femoral adipose tissue.
Yeah, that's a very interesting perspective. And I can see where you're saying like there's a little bit of a contrast, but what I really think is happening is that, you know, we think of femoral adipose tissue or femoral fat as a safe place to store fatty acids. As you say, it's healthier. And if you look at the correlations, when you have a higher amount of femoral adipose tissue, you actually have a decreased risk of death, right? Like it's an inverse compare. It's in contrast to if you have a higher waist circumference, you're going to have a higher risk of death, right? Now, what I actually think is happening is that the subcutaneous adipose tissue is becoming dysfunctional. Like, so it's not, it's in a, like it becomes such that it has there's impairment in some adipose tissue characteristics that it actually may impair fat storage in that depot. And as a consequence of the impaired fat storage in that depot, we can no longer store fat safely in that depot. And when you can no longer store fat safely in that depot, well, that fat has to be stored someplace. So where it's going is either in your upper body subcutaneous adipose tissue depot is a lot less safer, or it can be going into your visceral adipose tissue storage. So, or in and around your organs, right? So I feel like maybe what it could be is that a larger visceral adipose tissue depot maybe could be an indicator of the fact that you have dysfunctional femoral subcutaneous adipose tissue. And interestingly, in that study that was it was with Devu, they were looking at like a transcriptome profile of those who had upper body versus lower body fat. And they actually saw that in those who had upper body adipose tissue or upper body fat accumulation, that the lower body, there was actually dysfunction in the lower body or femoral subcutaneous adipose tissue, where they had genes that were associated with immune cell chemotaxis and inflammatory pathways that were upregulated. So this is in the lower body of those who had an upper body phenotype, indicating that perhaps it is a dysfunction of lower body phenotype. And then as we age, you know, I haven't really looked so much in terms of, like, we know that the femoral adipose tissue with, like, for example, post-menopause becomes somewhat dysfunctional as well. We start to accumulate more fat in your upper body. And then as a result, we have an increase in disease risk, right? In females, I should say. So I think that it's a very interesting question, and I think we really don't know enough about femoral subcutaneous adipose tissue. And a lot of the times what happens, and part of the reason that we get a lot of comparisons between abdominal subcutaneous adipose tissue and visceral adipose tissue, is that in surgery, it's relatively easy to get the two depots to compare them. And for us to do actually our femoral subcutaneous biopsies, we actually have to have people come into our center and we have to aspirate the fat from them. So we don't, you know, it's like an additional thing that you have to do, which we've been grateful to have participants be willing to do this, to look a little closer at their subcutaneous adipose tissue characteristics.
Thank you for your answer. And I know how it is complicated to have such a measure in adipose tissue as I did myself. And it was not easy to have participants agree for such biopsy. So thank you very much.
Yeah, exactly. It's partly why it's taken so long for us to get our results.
Mylène? avec le micro. Si possible, avec le micro. Ça peut nous aider.
Sorry, I was thinking you are in control of our micro. So very briefly, do you think, as you know, the metabolically healthy obese are protect against cardiometabolic disorder? But we also observe in the literature that when you are doing a diet restriction or exercise intervention, you deteriorate their metabolic profile. So do you think that people with a good inflammation profile using subcutaneous adipose tissue located in android or femoral, compare those with a high inflammation, will answer on the same wave using exercise or diet restriction to prevent obesity? Because in older adults, it's very controversial if we need or not to intervene to decrease the amount of obesity, because it could be more deleterious than beneficial. So do you have any clue if inflammation will be our guideline?
In terms of the exercise interventions?
Exercise or diet, yeah. Because in some case, in metabolically healthy obese, you deteriorate the insulin sensitivity when you intervene, without, even if you think you are protecting them against diabetes. So do you think that people with femoral adipose tissue, even if it's inflammatory or inflammaging, will be something that declining this amount will deteriorate the metabolic profile and not the improve, like the MHO?
So you're saying that with an intervention, you're in those who are in older adults, the insulin sensitivity decreases when you decrease adipose tissue.
It was in postmenopausal women. It was not in older adult. It was in postmenopausal, but metabolically healthy, but very obese, BMI over 40, if I remember correctly. It was done in Montreal mostly, so.
I mean, it's hard to say because it also depends on the length of time. Do you know what the length of time of that intervention was?
It was 3 months. Eleanor could confirm me, but I'm quite sure it was 3 months with a diet restriction or exercise, and both intervention decline the metabolic profile, so deteriorate the metabolic profile.
Oh, that's very interesting. I'm not really sure how to address that, because exercises and diet, of course, is thought to improve the metabolic profile, right? And so the fact is that you're actually having deleterious effects on the metabolic profile is somewhat surprising. One thing I do know is that exercise increases lipolysis. So I wonder whether or not the effects of the increases in lipolysis and the effects of what that might have to on the muscle, skeletal muscle, could have potential like lipotoxic effects in these individuals. But I mean, we've always thought of exercise as a beneficial and weight loss as a beneficial, even regardless of metabolically healthy obese individuals. Did you look at do you know that they looked at the body fat distribution in terms of in these individuals? And so this was in postmenopausal women that both had upper body and lower body fat distributions? Yeah. Oh, wow.
And we observed the same in Quebec during my PhD. This is what I was talking before. We had a deleterious impact of exercise after 12 weeks of brisk walking. So, and pretty the same, metabolically healthy postmenopausal woman with no drugs. So it was quite surprising, though I think that there is something that we maybe need to investigate a little more when we look at femoral adipose tissue in healthy versus not unhealthy participants. And the fact is, in Quebec, the BMI was lower than the people in Montreal. It was around 30, 35. So it's not, too high. So I need to go, but this is so interesting. I'm so sorry. I'll let you answer,
thank you
Milan. Thank you, Milan, for your question too.
Thanks for joining us.
Sorry. Go ahead, Sylvia.
Oh, no, it's okay. Go ahead.
Ok, je voulais vous proposer encore une ou deux questions si vous voulez, vu qu'il reste quelques minutes. I was like, if you have any question, we have 1 or 2 minutes more. If not, I have one. I have a really quick one. Is like, what's the role of physical activity in this? I have 2 questions, in fact. The first one is, do we know any difference between young and old adults in terms of like the quantity of fat mass on upper and lower body or abdominal? Because like you show difference men versus women, so I was curious about young versus old adult. And the second question was, what's the role of physical activity? Because like I'm not expert in this area, but when I see all the information and mechanism, I saw some overlap with some enzyme and things like that with related to physical activity, expending energy, and this kind of thing. Do we know the role of physical activity for mediating this and how it could help for the difference between upper and lower body fat mass? It's 2 different questions, sorry.
Yeah, so let me address your first question. Your first question in terms of, okay, if I understand your question correctly, we currently have a study that's going to be looking at aging versus obesity onset. So we're hoping to delineate the effects of aging and obesity onset to subcutaneous adipose tissue. And so we don't have the results for that yet, but we're looking pretty much at immune cells and fibrosis and what else was it? We have macrophage, fibrosis, and fat cell size were the ones. This is a study in collaboration with a group at Laval. And your other question in terms of exercise,
I would say more physical activity, the level of physical activity, whether the acute effect of exercise, like when people are regularly active, does it change those mechanisms you described?
Yeah, that I'm not sure of. I'm sure that there are studies out there, but I cannot recall what the actual results are.
Okay.
So, but thank you. That was a good question.
No problem. Si vous avez encore des questions, n'hésitez pas. On peut en prendre une dernière. A last question from the audience, if you have some. Okay. Thank you. Donc, on va pouvoir s'arrêter là. Thank you again, Sylvia, for this amazing presentation. That was really, really interesting. Et je vous souhaite à tous une bonne après-midi.
Okay. Thank you, everybody.